Greetings students, welcome to today's session of the Algorithmic Thinking in Bioinformatics course. I want to elaborate on the greetings by just greeting you either a good morning or a good afternoon or good evening or good night, depending on which time of the day you are watching this video, and a slightly different form of greetings compared to my earlier sessions has relevance to the topic that we are interested in today. We are going to look at certain regulatory proteins that have relevance to the kind of notion of time that our body maintains. So, what time of the day we are in, is it the day, is it the morning time, the mid noon or the night time, right. So, there are certain proteins that are involved in helping our body understand the notion of time, and how do these regulatory proteins regulate or control other genes to establish this notion of time, right. And then the way they do it is they bind to certain sequences, DNA sequences which are upstream of these target genes. And how do we find those sequences or motifs, DNA motifs, okay. So, that is the biological question that we are interested in today, and we would look at computational approaches to figure out these DNA motifs that other regulatory proteins such as these transcription factors involved in maintaining this notion of time, right, that these transcription factors bind to, okay. And so, these computational approaches will involve certain greedy approaches, but mainly randomized algorithms in the form of GIP sampling based algorithms, okay. So, that is what we are going to look at today. So, in terms of the context for today's session, we have seen several biological questions or clinical questions recently and their algorithmic solutions as part of different sessions of this course, and today will be no different. So, we will start with this important biological question related to establishing this notion of time that I talked to you about, and then how do regulatory proteins or transcription factors in general bind to DNA sequences to regulate other genes, okay. So, how do we find the sites or motifs in DNA sequences that a gene regulatory protein transcription factor binds to? So, how does it detect which subsequence of a DNA sequence that it should bind to, right. So, how will the transcription factor determine that, okay, and how do we find these transcription factor binding sites and motifs, okay. And then we will look at what is the associated computational problem. We can experimentally figure that out, but are there computational approaches to figure this motif or k-mer, specifically k-mers that are enriched in a set of DNA sequences that we know that a transcription factor already binds to, okay. And finally, we will look at this solving this problem using algorithmic solutions, mainly two approaches. One will be a word enumeration or a k-mer enumeration approach that is not really probabilistic, but based on some scoring scheme and optimizing that score, and then a probabilistic GIP sampling based approach that is a randomized algorithm for figuring out certain motifs and sites in a set of sequences that a transcription factor binds to, okay. And this flow of starting with a biological question and then moving on to a computational problem and then its solution is central to this course, and in fact central to bioinformatics, I would say. I want to say a small anecdote related to that. Yesterday in our campus, there was Professor Vijay Tiwari who was visiting us, and then he was interacting with students and moving around students and asking them for what are the problems they work on. These are bioinformatics students, and some of them started with a method and then went on to talk about what question that the method was supposed to solve, and then he was trying to reorient or refocus the student to start with the biological question and then see what method or tool was used to address. And for that he was saying that bioinformatics, there is a reason that bio is in front of informatics in the word bioinformatics. So, we always start with a biological question so that we keep our focus sharp, and then we look at of the different methods and tools that we have in our data science toolkit, what can be applied to address that particular biological question, right. And we will follow that same tradition here and move on to recall certain concepts, the biological concepts, and in order to formulate this motif finding problem clearly, okay. So, we have seen this gene regulatory network before. So, genes do not act in isolation, certain genes regulate other genes, they are basically transcribed and translated into proteins, right. So, the gene is transcribed into mRNA and then translated into this protein 1, and then later on that particular gene 1 through its protein product can regulate other genes such as gene 2 and gene 3, right. So, you can see here the arrow coming from gene 1 to gene 2 and gene 1 to gene 3. So, there is a single gene, gene 1, which encodes a transcription factor protein that regulates gene 2 and gene 3, and the way that it regulates, it is bind to some sequences that are upstream or downstream of these genes. So, in the gene 2 you have this upstream sequence and then this protein goes and binds there, right, and then it regulates this gene 2, and here the same protein or other copies of the protein can bind to this downstream sequence of gene 3, right, over here, and then it can regulate gene 3, okay, it regulate the expression of gene 3. What we are interested in is what specific site does this protein 1 bind to in front of gene 2 and in front of gene 3, and what is the pattern that you can extract from these sites, okay. And this is just 2 genes that I showed, but you can have multiple genes. You can have a transcription factor that can regulate multiple target genes, TG1, TG2, TG3 and so on. So, there are many target genes that a transcription factor can bind to, and then these can be figured out through certain experiments on which genes a transcription factor binds to, right. But once you have these genes, then each gene in the genome is going to be something like this, right. So, you have this target gene 1 and then you have some upstream sequence, and then you have this target gene 2 somewhere else in the genome, maybe this is in chromosome 1, maybe this is in chromosome 2 and so on, right, and then you have this upstream region and so on, where this may again be in chromosome 1, but somewhere else in chromosome 1 compared to TG1, and this is this upstream sequence, DNA sequence, right. And we want to figure out where exactly, maybe in this particular genome sequence it will bind, the transcription factor will bind somewhere in the beginning of the sequence, and the TG2 it may be binding somewhere in the middle of the sequence, and in TGN it may again be binding somewhere in the beginning of the sequence, right. So, there are different locations upstream of these target genes that the transcription factor can bind to, and those locations or sites are the ones that we are interested in, okay. So, we are given these different target genes TG1, TG2, TG3 and so on. These may be in different chromosomes, different locations of the same chromosomes, wherever it is. This transcription factor gene and the gene and the protein encoded from that gene, right, can now bind to different locations. Like I said, beginning of this, maybe end of this, and then beginning of this, maybe here it is the end of this. So, it could be binding in the beginning of TF1, the middle of the upstream sequence. So, these are all these upstream sequences of each of these genes in different chromosomes or in different locations of the same chromosome. So, this transcription factor can be binding to the beginning of this particular upstream sequence, in the middle for the TG2, maybe in the end for TG3, and then in the beginning for TGN, right. We do not know where exactly they are binding in, but that is the problem that we are interested in solving, okay. So, when you have a transcription factor, and then when you know the target genes that a transcription factor binds to, then you can extract all these upstream sequences, and then the problem is where exactly does this transcription factor bind to, and then based on where it binds. So, usually these wherever it binds, it is usually about, let us say 5 to 20 nucleotides in length, okay, where the transcription factor protein will bind to. I am just going to draw it like this blob, okay. So, each of these may be 5 to 20 base pairs in length, right, where it binds to, right. Each of these stretch of 5 to 20 base pairs, right, will have certain similarities, right, because it is the same transcription factor that is binding to. A transcription factor does not bind to a random string, any random string. It will have a certain preference that it likes certain DNA sequences that it binds to, and it will kind of stay away from certain other DNA sequences, right. So, now what is the pattern that is present in these 5 to 20 base pairs, whatever it is? For different transcription factors, they may have different preferences of lengths also, right. So, whatever this pattern that it binds to, right, whatever sequence it binds to in all of these n target genes, is there a certain pattern that we can discern from looking at these sites, okay, and that is called, this pattern is called the motif, and you can also think about it as a k-mer that is present with certain mismatches maybe in all of these sequences, right. So, we are interested in where exactly, which sites upstream of the target genes do the TFs, the transcription factors, bind, and then what is the corresponding binding motif or pattern at these sites, okay. And then to answer these questions, there are both experimental and computational approaches possible. The experimental approaches are mainly revolving around chip-seq. So, that is a very popular technique. There are other techniques as well, but chip-seq is a popular technique for discovering all the different sites in the whole genome where a transcription factor binds to. Okay. And it has two steps. One is chromatin immunoprecipitation, that is to identify from a sample, right. Let us say you grow cells in certain media, and then you extract those samples, and then you expose it to certain chemicals that will make sure that whichever DNA is bound to a TF are just fixed together, right. And then you remove all the other DNA, and you only look at the DNA that has bound to this transcription factor. Then you perform a sequencing experiment to figure out what this DNA is, what these DNA sequences are. And those DNA sequences in the form of reads, right, sequencing reads are then mapped back to the reference genome, and then wherever the reads are mapping in high numbers, that the transcription factor has bound to that part of the genome. Okay. So, that is the chip-seq experiment that will give you in a genome wide scale, right, all the parts in the genome where this transcription factor binds to. And then there are also other more focused approaches like these gene editing techniques, CRISPR-Cas9, where if you know a particular location in the genome where a transcription factor binds to, let us say from some earlier chip-seq discovery experiments, you can validate it by just editing those sites and then seeing if the transcription factor binding is lost. Okay. And so, that could be used for validating certain regions in the genome where a transcription factor binds to by just editing those regions and checking if the binding is lost. Okay. But these kinds of experiments, both these discovery and validation experiments, are expensive and time consuming, mainly because of the quadratic space of the possible interactions, right. Whenever you have two entities involved and you want to figure out the pairwise interaction between those entities, here in this case you have transcription factor and target genes. There could be many transcription factors, and then there could be many target genes for each of these transcription factors, and then so you just multiply the thousands of transcription factors with all the 20,000 potential genes it could target, right, in the genome. Then you have a huge number of pairwise interactions that you have to experimentally test, right. And so, experiment alone is not sufficient. So, but a combination of experiment and computational prioritization can greatly help map the space of transcription factor target gene sites, okay, where they are interacting. And knowing these sites can help us then later intervene and then prevent or enhance the regulation of the target gene by the transcription factor. So, what does this computational prioritization mean? So, we have a computational motif finding problem, which is you know all the TEGs that a particular TF binds to, right, but you do not know where it binds, right. So, you then extract all the upstream DNA sequences of all TGs. So, what do I mean by upstream, right? So, you have a target gene, you look at the 1000 base pairs that is just before the start of this target gene, right. So, that could be a starting point, that is 1000 base pairs upstream of the target gene. Similarly, you go to every other target gene that the same transcription factor binds to, and then get all these upstream DNA sequences. Then you can look at among these 1000 nucleotides that I have gathered from all of these target genes, where exactly is this 5 to 20 base pairs region that I am looking at, the site that I am looking at where this transcription factor binds to, right. And then once I find these sites, then I can look for is there any preferences in terms of the nucleotides that are present, there ACGT's sequence preferences that this transcription factor has to determine whether it wants to bind there or not, right. So, that is the corresponding motif or pattern or k-mer that we are interested in, and this is taken from this particular review article by Fan, Yuan and Lu, and they argue that motif finding is a very fundamental problem in quantitative or statistical modeling of biological data, and especially the use of probabilistic mixture modeling and then the associated EM algorithm have given really good advances in this problem of finding motifs in a set of DNA sequences. Okay. And there are largely 3 different approaches again that is described in this review article. One type of approach is word or k-mer based enumeration, right. So, you essentially go over all the particular k-mers for some k value between 5 and 20, right, and then you have some score that tells you whether a k-mer is really the kind of pattern that the transcription factor binds to in all these DNA sequences that are provided as input. So, you just enumerate over all of these k-mers, find the optimal scoring k-mer and then report it. Okay. So, this represents a motif as a specific consensus word or k-mer, and then this has been done in the 2000s. Okay. And there are these profile or probability based approaches, where you represent a motif as a profile, a probability profile, or in other words a position specific weight matrix. Okay. So, if you have your k-mer, k length is phi, then for each column you just express the first position, the second position, and so on till the fifth position. Each position you represent what is the preference for the transcription factor. Maybe a particular transcription factor will only bind to a phi mer if the first position is always A. Then the weight matrix will be 1 for 100% for A, and then for CGT it will be 0%, right. And then maybe the second position can be either a G or a C, then that will be 50-50, and then the other A and T will be 0-0, right. So, that kind of weight of frequency matrix you can have, and that matrix will be for DNA sequences. You have 4 letters in each position. So, you have 4 rows in the matrix, and then the number of columns will be the length of the motif that the transcription factor binds to. If it is a 20 mer, then you have a 20 cross 4 position specific matrix. Okay. And then these algorithms, like I said, as early as 1989, and then the EM algorithm was actually introduced in Lawrence and Rayleigh in 1990. So, the EM algorithm we have seen as a way to do inference and parameter learning of a mixture model, and then we saw how it is useful for k-means clustering. So, the same EM approach is also applicable to the motif finding problem, and Lawrence and Rayleigh introduced the EM algorithm to solve this motif finding problem as early as 1990, and there were other works also in 1993 and so on. And there are hybrid and other approaches where we have Liu, Brutlag and Liu in 2002, and then in a few other reviews and comparative analysis of different motif finding algorithms by Jensen et al. In 2004 and Tompa et al. In 2005. Those are all good references of the early works in computational motif finding. Okay. And very recently there are deep learning based motif discovery tools also being developed, which uses word embeddings or these kind of representations of these k-mers, and then trying to figure out can the embeddings or representations of the motifs that are discovered by these deep learning approaches, do they tell anything about the geometry of how the transcription factor bind to these motifs. Okay. So, those are some recent topics of interest, and so this is a very current problem with a very old history, and so this is a fundamental problem to look at for any genomic data scientist, if I will, right. So, if I have to say. And that is really what is being argued in this article as early as 2010 also, saying that this is a fundamental problem that gives you a view, a quantitative or statistical view of biological data, a rigorous statistical view of biological data. Okay. So, for all these reasons it is a good problem to focus on, a good statistical problem to focus on, and a good problem that any bioinformatician or genomic data scientist should be aware of and how it is solved. Okay. It will take several lectures to cover all of the methods here. So, we are going to focus on just 2 different approaches. One is this word or enumeration approach, the most common ones there, and then a Gibbs sampling based approach. We will actually look at an EM based algorithm in a later session, but for now we will just start with this enumeration based approach, one popular approach, and then another Gibbs sampling based approach, which is a Bayesian approach to doing this profile based motif discovery. Okay. That will be the plan for the rest of the talk. So, we will now try to make the computational problem concrete and also the algorithmic solutions of this problem concrete as well, of the 2 approaches that we are going to focus on. These 2 approaches are the ones that are focused on in the book by Compo and Pepsner as well, and then again, as always, we are grateful to them for sharing the slides to talk about this particular topic. Okay. So, how do we find regulatory motifs in DNA using these 2 approaches of motif enumeration and randomized algorithms. Okay. And to do that, let us just look at some real sequences, because we have been thinking in lines and boxes. Let us just make it concrete using examples. How do you look at a set of DNA strings, and then how do you look at patterns in those strings? Okay. So, let us say that we generate 10 random DNA sequences. So, each letter is ACGT, each position is ACGT with probability 1/4 each, and then you generate these 10 random sequences. Okay. And then you may want to implant a particular motif or pattern at randomly chosen positions. Okay. So, you have a bunch of A's and then followed by a bunch of G's, and you are going to implant it at random positions in each of these sequences. Okay. This is supposed to capture the upstream sequences of 10 target genes, right, that a transcription factor binds to, and then in those 10 target genes upstream sequences, the transcription factor can bind to this particular motif. It likes to bind to sequences which has a lot of A's followed by a lot of G's. Okay. But now the question for us is, as computational data scientists, we want to know where are these implanted patterns hiding, because this generative process is not revealed to us, right. So, this 10 random sequences are generated, and then the same motif is implanted randomly in these 10 sequences, but we are not told where they are being implanted. So, that information is removed and we are just given these sequences, right. So, how will you find out where they are being implanted, right. So, we know that because you have just seen the other slide, you know that there are these lot of A's followed by a lot of G's. So, a lot of A's followed by a lot of G's. So, there are these different locations where this particular k-mer keeps repeating, right. So, lots of A's followed by a lot of G's. So, you could use the frequent words or frequent k-mers problem, right. So, you are trying to look for in these set of sequences what is the k-mer that keeps on occurring, right, in every one of these 10 sequences. So, you can do that, and then you can figure out that for a certain value of k, if you already know some guess on your value of k, then you can look at certain k-mers that occur more often in these sequences compared to random chance. So, that is what we call as over representation or enrichment, right. So, which k-mers are enriched in these sequences or present more often than by random chance in these sequences, right. And so, we can look for k-mers that are most frequently present and then identify these. But then here is a twist to the problem. Let us say that I am not putting these motifs exactly in these sequences. You randomly implant them, but then you also add 4 random mutations at random positions wherever you have implanted. So, those are the ones that are shown in red. Okay. So, there is a lot of A's followed by a lot of G's, but then in these 4 positions you just replace an A with a G and then a G with a T here. Okay. In the second sequence, similarly, you have these 4 red positions where the motif, which is all A's followed by all G's, is now disturbed. Okay. So, you have a new problem now. Instead of finding a k-mer that is present in all these sequences, you want to find a k-mer that is present with at most 4 mismatches in all of these sequences, right. So, that is called a k comma d motif. It is a k-mer that appears in each sequence with at most d mismatches. Okay. So, this is this lot of A's followed by a lot of G's is a 15 comma 4 motif. So, there are 15 letters here, and then there are 4 differences that you are allowing in the worst case, right. So, a k comma d motif, a 15 comma 4 motif. So, this particular A is followed by G's is a 15 comma 4 motif for these set of sequences. Okay. And this then complicates the problem. Now, will the frequent words problem or the algorithm work, right. So, there are all these extra mutations that are being added, right. Now, where are the implanted patterns? Now, it's no longer easy as easy as before, because we are asking again, we have to go through all the k-mers. If it is 15 mers, we have to go through all the 15 mers. If you know somehow guess the value of k as 15, then we have to go through all the 15 mers, and we will have to figure out which of these 15 mers are present with at most 4 mismatches in all these strings, right. And and so, there is it may not be as easy as the frequent words problem because of all these mismatches. Okay. The frequent words algorithm may not be able to find out, especially when the number of mismatches start increasing, right. When the number of mismatches starts increasing, then there may be many k-mers which are present with at most 4 mismatches in these set of sequences, and then it will start getting more and more difficult to identify the truly implanted motif. Okay. And now you may be wondering, why would a biologist care? This again I go back to the comment I told about what Professor Vijay Tiwari was saying. So, the bio comes before bioinformatics, before informatics in the term bioinformatics, right. The bio comes first. So, let us just also kind of make sure that whatever we are dealing with, these random sequences and these implanting and other things have relevance to the biology that we are looking at, right. And especially I started this lecture with the greetings on the relevance of time. Okay. So, let us just look at some genes that are important for maintaining the notion of time, and that is this clock gene. Okay. So, do we have a clock gene? The idea is that our schedules are controlled by a molecular timekeeper called the circadian clock, right. So, this is the day night rhythm that we maintain. So, we should know what is the what time of the day we are in to have a proper sleep schedule, the proper eating habits and everything that is required for proper sustenance and life, right. And so, are there genes that are actually maintaining this internal circadian clock, right. So, specifically, how does each cell in our body know when it should decrease or increase gene expression of circadian genes, right. So, when we are hungry, we need to increase the expression of genes that will produce acids in the stomach, so that the food can be digested, right. And but when we are sleeping, we do not need that, so those genes have to be switched off. And so, there has to be like the different cells in the body needs to know this notion of time, and then regulates a gene expression or activity accordingly, right. And so, how does this happen at the molecular level, which genes are involved, okay. We are not going to look at the clock genes in humans, but let us just look at some of the plants, right. So, plants need to change gene expression of 1000 plus genes, right, and there is a very clear need for them to know when there is sunlight and when there is night, because they need photosynthesis to generate all the resources that they need to live, right. So, the plants need to change the gene expression of about 1000 plus genes, right. So, photosynthesis, flowering, it needs to figure out when there is frost resistance and so on, right, and it needs to be able to switch between day and night, right. So, and this is a in terms of flowering example that is showing, right, this is a night blooming cereus, so it only blooms at night. So, it needs to, the cells need to know when to start blooming, right, the genes have to be regulated in that way. Sunflower follows the sun, I mean it just follows from the name of these flowers itself, right. So, who are the molecular managers telling genes to switch expression on and off in billions of cells that are inside each organism? And you will be surprised to hear that for plants, at least there are 3 key genes. There may be several other genes involved, but at least to simplify things, just 3 genes call the shots. These are these 3 genes, CCA1, LCY and TOC1. Okay, these are kind of master regulators of all the other genes that are involved in maintaining this internal notion of clock or time, right, the circadian rhythm. And these 3 genes are essentially transcription factors, right. So, these are the regulator genes, right. So, these genes encode proteins which are the transcription factor proteins, and that in turn control to several other target genes and then tell them or regulate their activity, so that the night and day functionalities of the plants are maintained, right. And so, and these transcription factor binding sites, like I have said, are the short DNA fragments in the upstream regions of all the target genes that are controlled by these 3 different transcription factors, okay. And that is shown, kind of the structure is shown, these are transcription factors and they are binding to this DNA sequence, and this DNA sequence shown as a double helix is the one that is upstream of these target genes. And a more filled representation is shown here, where these 2 transcription factors are now binding to the upstream region of a target gene, okay. For our session here, we will mostly be looking at transcription factors, one transcription factor binding to the upstream region of several target genes, okay. That is what is shown here. You are focusing on only one of the transcription factors, CCA1, and then you have these different genes in different parts of the same chromosome or on multiple chromosomes that are all kind of shown together as one long string, the genome, right. These genes are located in different parts of the genome, and then there are these copies of this transcription factor protein CCA1 that is binding to these different regions upstream of these genes. So, the red color is the gene, and then the purple color is the motif that is upstream of these genes that these transcription factor protein copies bind to, okay. But then the idea is, how will it know where to bind, right, because within a cell, if you just imagine what is happening within a cell, it is a huge mess, right. You have this nucleus, and then you have this whole genome. In our human, we have 3 billion letters that is all compacted into chromosomes, and so these CCA1 proteins, there are a few copies to a few thousand copies, let us say a few tens of copies or hundreds of copies or thousands of copies, whatever it is, each of these copies of this CCA1 protein has to wade through this highly trafficked region within the nucleus, and then it will have to figure out where the target genes are and then bind to those sequences that are upstream of these target genes, right. And so, how does it figure out where to bind? There must be some hidden messages in these DNAs that tell it where to bind, right, and then those hidden messages or motifs are what we are interested in figuring out. We are interested in figuring out computationally, right. So, these regions tell CCA1 where to bind, right, and then if you look at expanding out these upstream regions of these genes, right. So, there are these motifs that are being shown here, right. So, the hidden messages here, the motif is like lots of A's followed by TCT, right. So, this is the preferred motif for CCA1 transcription factor protein, right. So, CCA1 protein likes to bind to DNA sequences which have a lot of A's followed by TCT. It can permit some mismatches, those are shown in red colors here. So, there is a T and a G here instead of an A and a A. Here is a C here. So, there are some mismatches that it can tolerate, but it generally prefers DNA sequences where there is a lot of A's followed by TCT, and that is where it binds very strongly. To other sequences it will bind weakly, okay, the affinity of binding will be weak. So, that is the hidden message or the motif that this transcription factor binds to. But then often we do not know exactly where it binds, right, and then so we are just given these sequences and we are asked to find out what is the motif among these or the sites in these sequences where this transcription factor binds, okay. And that really is the computational motif finding problem, right. You are given a set of genes, you know that all of them are bound by the same transcription factor called CCA1 here, right, or any other transcription factor of interest. You are given these upstream sequences and you want to find a motif of a certain length k, a k-mer where the protein binds to, okay. So, hopefully this puts the biological question front and center and then puts bio in front of informatics in the term bioinformatics, okay.